Very much. So, good morning. The topic of my current research project concerns the generalizability of age-related differences on brain structure on the one hand and cognitive performance on the other hand. Now, during normal aging, cognitive performance declines. This is especially seen in functions such as attention and executive functions. Well, if we look at verbal knowledge, we see that this remains relatively stable across adulthood. Looking at brain structure, we see that from early to late adulthood, so here in this example from 20 to 80 years of age, there is a decrease in cortical thickness, and this is most pronounced in the frontal as well as the temporal lobe. However, if we compare these results to another study from our research group that conducted age-related differences in cortical folding in an older adult population, we see that the results are a little bit different. So here, in terms of cortical folding index, we see that there's no age-related differences in the medial prefrontal cortex anymore, here shown in green. And the most pronounced decrease would be in the medial parietal lobule. So when we compare the two studies, we see that they are quite different. And we also see that there's a limited generalizability in brain structure, but also in cognitive functions. And this is due to differences in two things. And that's first, the methods that are used. So here, for example, cortical thickness versus cortical folding. And second, due to differences in sample characteristics. So, for example, here in this example, we do have different age ranges. But why is it important to actually generalize results from one sample to the whole adult population? This is because in the current time, big data science is growing and growing. And this ranges from the large imaging consortia, such as Enigma, which pool data from independent samples together, ranging over multi-center studies such as the UK Biobank, but also the German national cohort that use the same imaging protocols and the same imaging hardware. And on the other hand, we do have the more specific but smaller single-center studies. And the aim of the current study was a cross-validation of age-related differences in cognitive abilities and in brain structure, and therefore we used two large single-center studies to verify whether data pooling might be useful. Now, let's have a look at the methods. First of all, the two samples. On the left side, you see the so-called LHAB study in orange. That's the longitudinal healthy aging brain study conducted at the University of Zurich in Switzerland. On the right side, you see the study of 1,000 brains that was conducted by our research group in the research center Jülich in Germany. And we first matched the two samples regarding their age and gender distributions, finally resulting in 228 subjects per sample, ranging from 65 to 85 years. And we had in each sample 114 males and 114 females. Now, in the first step, we then did the sample comparison, and we did that with respect to the demographics, for example, education, with respect to cognitive performance, and here we looked at the domains of attention, executive functions, and language functions, and with respect to brain structure, and here we used cortical thickness as one measure of brain structure, and we extracted this for each hemisphere and for all the parts of the default mode network, as shown in the lower right corner. And then in the second step, we did a cross-validation of our age-related differences in cognitive performance and in cortical thickness in brain structure. Now, let's have a look at the sample characteristics first. So if we compare the two samples, we see that the LHAB sample, shown in orange, showed more years of education and had a higher physical well-being. In line with that, they also showed a higher or better cognitive performance in all the different tasks that we assessed. In contrast, looking at cortical thickness, we see that subjects involved in the 1,000 brain studies had higher values of cortical thickness, and that was not only true for the two hemispheres, but also for all parts of the default mode network. So in general, we see that the two samples were quite heterogeneous. So now let's have a look at the cross-validation of our age-related differences. And here we start with cognition. In the first example, our reasoning task, we see that with increasing age, shown on the x-axis, there is a decrease in performance. And that is true for both studies, for the ALAP study, shown in orange again, and for 1,000 brains, shown in blue. And if we compare the two correlation coefficients, we see that they do not differ significantly from each other. So we have similar slopes. That was not only true for the reasoning task, but also for all the other cognitive tasks we assessed. So in the upper row, you see tasks such as processing speed and concept shifting. That was measured as time in seconds, so older people need more time to complete the task. There's a performance decline. And in terms of vocabulary, we see that in both samples, this remained relatively stable across the older ages. What about brain structure? Looking at the default mode network and starting with the anterior part of the default mode network, here shown in green, we see that there were no age-related differences in cortical thickness in none of the two samples. In contrast, if we look at the posterior parts, here shown in blue and in red, we see that there were age-related differences in cortical thickness for both studies. And here again, the slopes were similar. So first of all, we could here show that we have a structural correlate for functionally established aging theories, such as the posterior to anterior shift in aging. And second, what we can conclude from this is that we found similar slopes of age-related differences in cognitive abilities on the one hand and cortical thickness on the other hand in our two samples. So if we see these results, the question arises, why would we not always pool data together to increase sample sizes and increase statistical power? And we try to answer this question by afterwards exploring effect sizes. So in this example, we pooled the data from the ALAP study and 1,000 brains together, and we assessed the relation between, in this example, our performance in our reasoning task and different covariates. And if you have a look at the green line, you see the results from the pooled data analysis. So in this example, age and education seem to explain most of the variance in performance in this task. In contrast to that, if we look at the ALAP study only, age seems to have the most important effect on performance in this task. And then, looking at 1,000 brains, we see that education might be the most interesting factor here. So if we compare the three analyses, we see that if we use pooled data analysis, we can extract general trends in terms of cognitive performance and which factors influence this performance. However, if we have a look at single-center studies, we might be able to extract all the particularities of specific study populations. We did not only do that for the reasoning task, but also for other tasks. For example, in the vocabulary task, we see that education might be an important factor, especially in 1,000 brains. And looking at processing speed, I think we all see that age might be the most important factor here. We also did it for brain structure. On the left hand, here in green, again, you see the interior default mode network. Age do not have any specific or important effects. Physical well-being might be an interesting factor in terms of cortical thickness differences. On the other hand, looking at the right lateral posterior default mode network, we see that for all the three analyses, age was the most important factor. So, what can we conclude from the current results? First of all, we had quite heterogeneous samples. We had a higher cognitive performance in the ALAP study. They might have a higher cognitive reserve. On the other hand, 1,000 brains had higher cortical thickness values. However, I here have to mention that the two samples were independent, so they used different MR scanners, and the differences in cortical thickness values might be due to scanner differences. Then second, we found similar slopes of the age-related differences in cognition and in brain structure. So we can say, yes, we can generalize results if we use the same methods and the same age and gender distribution. And lastly, we used single versus pooled data analysis, and If we have a look at the pooled data analysis, we can extract overall trends. However, if you want to look at population-specific particularities, single-center studies might be very useful. So in terms of big data science, we can say that we here nicely showed the trade-off between data pooling, that is useful to increase statistical power, and on the other hand, separate analyses that might be rather useful to cross-validate results. And with that, I would like to close my talk. I would like to thank my supervisors and the supervisors of the two studies, 1000 Brains being Svenja Kaspers and Katrin Amunds, and supervisors of the ALAP study being Susanne Meria and Lutz Jenke. Of course, all my colleagues, the team Connectivity, and you for your attention. Thank you very much.